Greetings to you all, ladies and gentlemen. I am Benjamin Azuakole from Ghana, specifically based in the University of Ghana in Accra. I'm happy to partake in the 5th Annual Congress on Plant Sciences and Biosecurity, and I will be talking on the characterization of Fusarium and Neocosmospora species associated with crown rot and wilt of African eggplant. My talk will throw more light on what the African eggplant is and progress to the results and conclusions of my study. The African eggplant, as seen on the screen, is the close relative of the European eggplant. That is the eggplant that is popularly referred to as Beringia eggplant or aubergine. The African eggplant is the most cultivated and the most consumed in the African continent. The leaves and fruits contains high amount of proteins, carbohydrates, iron, and then vitamins. It has also been shown to contain lots of bioactive compounds that have been shown to reduce high blood pressure. The crop is one of the critical indigenous crops that is strategically targeted for achieving food security and economic development in Africa. This is because it is relatively affordable and is available to the marginalized groups in Africa. It is also known to be drought tolerant and is being found to resist a lot of soil-borne diseases, including those caused by Ralstonia and verticillium. Unfortunately, the African eggplant is susceptible to a vascular wilt disease of an unknown cause. Historically, very little research has been done on the African eggplant in comparison to the European eggplant, where Fusarium oxysporum forma specialis melongena has been found to induce wilt symptoms. Some African eggplants have been shown to completely resist this pathogen, and for this reason, these resistant cultivars have been deployed for the control of wilt diseases in the European eggplant. Among most African eggplant researchers, it is mainly believed that it is this same pathogen inducing wilt in the European eggplant that causes the wilt of the African eggplant. And this assertion or this claim has actually not been supported by any research evidence. Not knowing the wilt causing agents of African eggplant has been a source of worry for many African eggplant farmers, and it has also made it very difficult to control the disease. As a result of this study was conducted to identify the prevalent fungal pathogen causing wilt of African eggplant in Ghana. That is to aid the development of specific management strategies. We hypothesize that a distinct fungal species other than Fusarium oxysporum forma specialis melongena causes the wilt of African eggplant. Specifically, the study sought to determine the incidence of wilt in major African eggplant growing communities to accurately identify and then characterize the associated disease causing agent, and then to confirm their pathogenicity on the host. So to achieve these objectives, field surveys were conducted in 2021 and then in 2022 across 21 major African eggplant farming communities that are located in the southern belt of the country. The southern belt is is the major area where the cultivation of the crop exists. So out of the 21 communities in southern Ghana, 36 farms where agriculture extension agents or growers reported disease incidents were studied in total. From these areas, the plants were assessed for disease incidents, and then samples were also taken and then sent to the laboratory for further analysis. So under laboratory conditions, a total of 274 suspected fungal agents were obtained from 121 samples collected from the field. The recovery of the fungal agent was conducted on potato dextrose agar. Followed by that, a preliminary morphological study was conducted to eliminate the duplicate isolates from each farm, and this left behind 53 representative isolates for the diverse fields that were surveyed in 2021 and 2022. The study aimed to do a detailed characterization of only the fungal species or the fungal isolates that were pathogenic on the African eggplant. For this reason, the 53 isolates were inoculated on African eggplants, and then subsequently 36 isolates were found to be pathogenic. These 36 that were found to be pathogenic were subsequently subjected to a multiphasic approach of identification that has been proposed by Cross et al. In following the polyphasic identification method, the cultural traits of the different fungal isolates were determined on PDA, and subsequently the spore traits were observed under a compound microscope. This was done after culturing them on SNA. The morphological identification was complemented with robust identification methods such as multi-gene sequencing of different barcodes, including the beta-tubulin region, the ribosomal polymerase beta-2 region, and then the TEF-alpha region. The sequence products obtained were blasted in GenBank, and then subsequently a phylogenetic analysis was conducted to determine the evolutionary relationship between the present isolates and then those obtained from GenBank. Now to the results section. Under field conditions, two distinct symptoms were observed. The first was plants that were wilting without any basal stem rot or lesions. For this category of plants, they displayed typical chlorosis and then wilting. And then in severe cases, the plants defoliated their leaves. And then also the fruits immaturely ripen on the plants. If you cut the stems of these plants into two halves, we notice the discoloration of the tissues in between the bark and then the hardwood, and these are typical indications that the organism that is involved is an organism that disrupts the vascular tissues of the plant. Further, we notice the outgrowth of the fungi or the suspected fungal agent between the bark and then the hardwood. That is, if you cut the stems horizontally and then leave the plants to stay overnight. The next category of symptoms was plants that were displaying typical wilt symptoms but also had rot lesions around the base of the stem. These rot lesions under humid conditions, we noticed that there was the fungi produced its signs, or there was a physical presence of the fungus on the basal stem regions. And this led to us asking as to whether the primary route of infection in the wilting plants in Fig 4 were through the roots or the stems. And then also we asked if the same fungal isolates were involved in inducing the crown rots and then also inducing the wilt symptoms. Table 1 shows the prevalence of the disease in the different communities and fields that were surveyed in 2021 and 2022. In all, we noticed that the disease incidence and severity was higher in the eastern region and then in the western region. These two regions constitute the deciduous rainforest and then the evergreen rainforest. The rainforest zones in the country are actually the places where oil palms are cultivated, and African eggplants too are also cultivated alongside. So there is a possibility that diseases that affect the oil palm may possibly jump and then also affect the African eggplant. Overall, it was noticed that the average disease incidence was around 33% in all the areas that were surveyed. Table 2 shows a summary of the morphological traits obtained for the different isolates. In all, we noticed that the isolates belonging to Fusarium elidis and Fusarium fragicuri showed the maximum growth rate on PDA compared to the Neocosmospora species. On the other hand, the spore length for the Neocosmospora species were relatively larger than those obtained for Fusarium fragicuri and Fusarium elidis. So the image displayed here illustrates the distinct phenotypes that were obtained for the different fungi species. A and B illustrates Fusarium elidis and Fusarium fragicuri respectively, with almost red pigmentations produced in culture. Whereas for the Neocosmospora species, we notice the production of brownish to yellowish colorations in the media. Their spores, as seen here too, are relatively larger compared to those of the Fusarium group. So the phylogenetic analysis also confirmed the identities of the various fungi isolates. It placed them into two distinct groups, one being the Fusarium group and the other being the Neocosmospora group. So the tree depicted here shows a truncation of the original phylogenetic tree, and is specifically for the Fusarium group. It was observed that 14 of the isolates grouped specifically with Fusarium elidis obtained from gene bank, and the grouping was supported by high bootstrap support values, close to 99%. Similarly, the single isolate of Fusarium fragicuri also grouped with its members with high bootstrap support values. Further, the truncated tree for the Neocosmospora group showed three distinct groups, including the Neocosmospora falciforme, Neocosmospora suetoniana, and then Neocosmospora solani. All these groupings were supported with high bootstrap support values. In terms of the number of isolates, N. falciforme and F. elidis had similar isolation frequencies. However, when we look at it in terms of the different fields that were infected, then F. elidis had a wider distribution compared to N. falciforme. In terms of the type of symptomatic tissues from which the isolates were obtained, the data showed that isolates obtained from F. elidis were exclusively associated with plants displaying only wilt symptoms. In contrast, all three Neocosmospora species, together with F. fragicori, were more often obtained from plants displaying wilt symptoms due to crown rot. The data suggests that F. elidis and F. falciformis are the most dominant fungal species prevailing in the areas that were surveyed. In addition, the Neocosmospora species induce typically crown rot symptoms, and probably the wilt symptoms that were observed were the secondary symptoms. Pathogenicity of the different fungal species was confirmed by inoculating young seedlings with spore suspensions prepared from the different isolates. The inoculations were performed mainly on the roots of the plant. 3 weeks after incubation, we observed that plants that were treated with Fusarium elidis displayed the typical symptoms of wilt as compared to the other 4 species that were obtained. They showed symptoms of reduced growth but not typical wilt symptoms. Specifically, as shown in B, the plant treated to F. elidis wilted from the top without any crown rot symptoms. The figure C illustrates plants that were treated with Fusarium fragicori. The plant showed yellowing and reduced growth, but not the typical wilt symptoms. Wilt symptoms only emerged in the Neocosmospora species only when there was evidence of crown rot at the base of the plant. D represents N. falciformis, E represents N. sutoniana, and F represents N. solani. For N. solani, we notice that it produced the teleomorph, on the plant, and these are images that shows the ascus, and then this is the perithecia for the species N. solani. Table 3 shows the severity of wilt on 3 African eggplant genotypes, and the data show that F. elidis induced severe wilt symptoms on the 3 African eggplants compared to the other Fusarium species that were obtained in the study. To further distinguish between the wilt-inducing pathogens from the crown rot-inducing pathogens, we inoculated the stems with mycelia plaques from the different fungal species. We observed that the Neocosmospora species induced the most severe symptoms on the African eggplant stems, whereas the F. elidis induced the least severe symptoms on the African eggplant. The bar graph shows the severity of crown rot, with F. elidis inducing the least symptoms, whilst N. solani induced the greatest lesions on the stems of the plant. In conclusion, the study showed for the first time that wilt of African eggplant is caused by Fusarium elidis, whilst Fusarium fragicori, N. falciformis, N. sutoniana, and N. solani contributed to wilt by primarily causing crown rot symptoms on the plant. F. elidis and N. falciformis were the dominant pathogens causing wilt and crown rot respectively on the plant. The findings offer valuable insight for developing targeted disease management strategies to control Fusarium wilt in African eggplant. Future studies should focus on screening a broader range of genotypes to identify sources of resistance to Fusarium elidis, which is also known to induce wilt on oil palms. I would like to thank the Danish government for funding my PhD studies as well as supporting the Fruit Bunch project. I also thank my supervisors, Dr. Francis Collison, Professor Ofe, and Professor Sabin for their continued support and encouragement. Thank you for your attention.